Lesson 7, Cellular Respiration. Cellular respiration is the oxidation of fuel molecules. Many different molecules can be used for fuel, but our primary fuel is the sugar glucose. And so we are going to be examining the oxidation of glucose to extract the energy. We introduced this concept back in the last lesson. This is the summary reaction, the simplified reaction for the cellular respiration of glucose. C6H12O6 is glucose plus 6 molecules of oxygen. Those would be the reactants. The end products are 6 molecules of carbon dioxide plus 6 molecules of H2O. And the cell can net up to 38 molecules of ATP throughout these reactions. This is a multi-step process. Again, what we're looking at is just the simplified summary reaction. Remember, oxidation is the loss of electrons. Reduction is the gain of electrons. In biological reactions, many times that electron is accompanied by a proton, and so this is hydrogen. So if we look at our reaction, if we strip all of the hydrogen away from that glucose and pass it over to oxygen, and then look at the other side of the reaction, and you can clearly see the products are carbon dioxide and water. This is the highly exergonic pathway, and so as we are doing these oxidation reduction reactions, the cell can take little bits of energy and ultimately produce all of these ATP molecules. Again, this has to be a multi-step process. We're going to take little bitty bites of energy from these fuel molecules, and so it will involve the movement of electrons, and they are going to be carried by 2 electron shuttles. These molecules are NAD and FADH. These we can think of as little electron shuttles. They're going to participate in some oxidation reduction reactions, and ultimately they will pass these electrons from the glucose to the oxygen. So here is how the NAD might work. If we start down at the bottom with NAD, it can be reduced. It can gain 2 hydrogen atoms. That's going to be 2 protons plus 2 electrons, and so it becomes the molecule NADH, and then that NADH can pass those. It can be oxidized, and it can pass those electrons off to another molecule. Oxidation of glucose, the cellular respiration, is going to occur in 4 different phases. The first one is glycolysis. That's followed by something that's called the preparatory reaction, sometimes the transition reaction, or the formation of acetyl-CoA, several different names for that reaction. And then the citric acid cycle, and lastly the electron transport system. And this is a diagram that we will refer to a number of times throughout this lesson as we are going through these different phases, beginning with glycolysis, the preparatory reaction, the citric acid cycle, and then the electron transport chain. Again, this is a number of little small oxidation reduction reactions, and so those high energy electrons, as they are passed from one molecule to another in these redox reactions, some of that energy from those electrons is harvested for the synthesis of ATP. The first step, glycolysis, we'll just summarize glycolysis. It's 10 different reactions. These occur in the cytosol, or the cytoplasm of the cell, so they are outside the mitochondria. And the name glycolysis means sugar breaking. Glyco refers to glucose, lysis is to break. And so during these 10 reactions, this 6-carbon glucose molecule is broken down into 2 3-carbon molecules. These 3 carbon molecules are called pyruvate. So one glucose is split into 2 pyruvates. This requires some energy investment initially. The cell has to spend 2 ATP molecules to get this started, but then it gets 4. And so the net yield of glycolysis, as far as energy, 2 ATP molecules are gained, and also 2 NADH molecules are produced. Now remember, this NADH also represents a little bit of energy that has been harvested. This was initially energy that was in the bonds of the glucose, and now that energy has been passed over as the NAD has been reduced. Before we move on, a little bit more about these pathways. Glycolysis is a totally anaerobic process. It does not require oxygen. Glycolysis can be completed without oxygen. If, following glycolysis, oxygen still is not available, then the pyruvate must be fermented. Fermentation is an anaerobic conversion of pyruvate. It does allow the cell to get some additional NAD and NADH oxidation reduction reactions to be completed. Fermentation can produce ethanol in some organisms. So yeast, for example, can ferment the pyruvate and form ethanol. So this is the reaction that we take advantage of when we're making beer and wine. In muscle of organisms such as ourselves, the end product of fermentation is lactate or lactic acid that can build up in muscle if we have vigorous activity and we're not getting enough oxygen delivered to those muscles. Fermentation is very inefficient because we do not make any additional ATP. We get those net 2 ATP molecules from glycolysis, and there is no additional ATP produced. It can be advantageous because it will allow some cells to live at least for a short period in the absence of oxygen, and it's also very quick. So if we need a little bit of energy very quickly, we can follow this anaerobic pathway. But this is not the preferred pathway for most organisms. What we would prefer is that following glycolysis, oxygen is available. And if oxygen is available, then the pyruvate, which is the product of glycolysis, is going to feed into that preparatory reaction. The preparatory reaction feeds into the citric acid cycle, and then the citric acid cycle feeds into the electron transport system. So this is the aerobic pathway. It is aerobic because oxygen is required, and the oxygen is required down in the final step. The last thing that happens is called the oxidative phosphorylation, where we convert ADP back to ATP. This is where the bulk of the ATP is produced, and this does require oxygen.